Okay, guys, you got through review one. Welcome to review two, the neuron in the brain. You got through the kind of slow research history part. Now we're into the biology heavy content. Remember, this one is all about memorizing brain structure, neuron structure, and its function. Flashcards are very valuable with this content. So let's get into it. First thing we're going to talk about is the lovely neuron. Now remember, you guys just need to know the basic neuron, but there's a whole lot of moving parts to this section. So first thing we'll talk about is my favorite, the glia cells, that no one else seems to be as excited about as I am. But remember, glia cells make up the bulk of the brain. This is what we're normally talking about when we talk about gray matter. So you have that cerebrum, this kind of eighth of an inch thick network of neurons that kind of almost fits like a swimmer's cap over all of these glia cells, and the glia cells kind of plump it up. But newer science has shown us that these glia cells will actually spark neural transmission, and in some cases they actually push the transmission before the neuron even gets excited. You see this in cases of things like fibromyalgia, where it's overexcited cells that actually kind of spur the pain message on. So the glia cells do this. The glia cells also do things like specific glia cells like the Schwann cells. They form the myelin sheath. They also help move nutrients in and out of the cells. They remove waste products. So not only are they structural kind of support cells, but they are literally environmental support cells for the brain. Now into the neuron, and I will show you a slide of a neuron again on the next kind of slide of this, but know the basic structure of the neuron. Go back and look at a diagram. Will you get a diagram? There's a possibility. So you need to know what the parts do and where they're located. First thing that we'll talk about, we're talking about the cell body or soma. So that's where the nucleus is going to be located. When we talk at the cell body, this is where you're going to have that postsynaptic potential. So after the synapse, this determines whether or not the cell has the potential to fire. And this is a graded potential, meaning that there has to be enough excitement, enough activity to start the action potential, or there is not. Those are the two options. The nucleus is going to be in the cell body. Surrounding the cell body, surrounding that soma, are going to be the dendrites. The dendrites, think of them as kind of branches or arms and extensions. Now, these arms don't technically catch. Remember, we talked about hot potato. They will actually receive like a high five stimulus, and that stimulus will then get calculated by the cell body. The dendrite's entire purpose is to have a receptor site, to accept a neurotransmitter, to get stimulation from it, and then to release the neurotransmitter back into the synapse. It does not absorb them. It literally high fives them. It gets the stimulus and then it releases them back into the synapse. Traveling past the cell body, you have a very long kind of spindly extension that comes out and then branches at the ends, and that's going to be the axon. The A's always go together. The axon has the action potential. So it's within the axon that we actually have the electrical charge start. And this is where we're going to have sodium and potassium ions moving in and out of the cell body. It's going to change the polarity from slightly negative on the interior to slightly positive. And that change in polarity is going to cause a fluid-like motion to move down the cell body, eventually causing the electrical charge to kind of body surf until it hits the very end of the axon at the axon terminals, pushing neurotransmitters to the edge of the cell body, causing them therefore to rupture and spill out into the synapse. Surrounding that axon that has the action potential is the myelin sheath. And the myelin sheath is a fatty tissue that surrounds the axon a lot like a plastic coating on an electrical cord. It condenses the message and moves it quicker down. So remember when we talked about nerve fibers and we talked about the fact that we have some myelinated nerve fibers and some that are unmyelinated, especially in our pain response. The myelinated ones are the ones that tell you, hey, stupid, stop it right now, it really hurts. The unmyelinated give you the throbbing message that just says, hey, remember you hurt this a while ago, maybe go easy on it. So myelin sheath causes a quicker sending of a transmission. Now, do not confuse that with intensity of transmission. Remember, your cells fire with the all or none principle. They either fire completely or they don't. Now, if you have a stronger response, it's not that it's firing stronger, it's firing more rapid. And that's that difference between a fire, 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 or a fire, 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 fire. One is going to be much more intense. We don't really talk about the nodes of Ranvier, but these are those periodic gaps within the myelin sheath, and it allows, again, for the ions to flow in and out. Terminal buttons are at the very end of the axons, just like the terminals at an airport are at the very ends of the airport. So these terminal branches, terminal buttons, they are the housing ports for your neurotransmitters. Okay, so just keep that in mind. Neurotransmitters are housed at the end of the axons. This is their home, so when they go out and they visit their dendrites, they come back home. They go back into the process of reuptake, back into the terminal buttons, back into those vesicles. Keep all of that in mind. Just like you would never leave your key in a lock, the lock is the dendrite, the key is your neurotransmitter, the key goes back into your pocket, make your pocket your axon, your terminal button. Resting potential. When a cell has the ability to fire, but it is not firing yet, it hasn't received stimulation, it's at resting potential. The big thing that you need to know about resting potential, the interior of the cell is slightly negative, the exterior is slightly positive. That imbalance causes the potential for the change. So that's where your potential energy comes. With the action potential, enough stimulation has been gathered by the dendrites and transmitted to the cell body where the postsynaptic potential creates an excitatory effect. The excitatory effect then spurs the action potential. The action potential is the electrical charge within the axon. A's always go together. Action potential, axon, all or none. When the action potential fires the electrical charge, this is the electrical part of the electrochemical kind of cycle here. When it fires the electrical charge, it will either fire it completely, 100% at full strength, or it will not fire it at all. There is no in between. So the all or none means that once it meets its threshold, it completely fires. Now, I always use the analogy of a gun. You can pull a trigger back as slow as you want, but that bullet comes out the exact same speed. That's the all or none. You pull it back hard, you pull it back soft, it's going to fire the exact same way. But if you're firing a revolver and you go fire, 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 you're only going to do so much damage to the target that you're shooting. If you're firing a machine gun and it's going fire, 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 you're going to shred that target. So again, the intensity is the rate of firing, not the strength of firing. Once all of this happens and you have neurotransmitters that have spilled out into their synapse, and the synapse allows that fluid-filled gap for those neurotransmitters to go over to the dendrite. They stimulate the dendrite. The cell body in the next cell starts its job of determining whether or not there's enough stimulation, whether or not it should start its own action potential. Those neurotransmitters are then released from the dendrites. They go back into the synapse. They go through a process of reuptake. Reuptake meaning that they go back into the axon. That's their home. They go back into the vesicles. The entire cell now has to reset. Sodium and potassium ions have to go back to the correct distribution prior, so it's slightly negative to the inside, slightly positive to the outside. This entire thing is the refractory period. During the reset, a cell cannot fire again. It needs to take a break. It needs to get everything back in order, otherwise it will not fire properly. And you guys have heard me use the term synapse here several times. Synapse, fluid-filled space that allows for the transmission of information between cells. And again, when we're talking about transmission of information, we are talking about neurotransmitters. Then we get into our neurotransmitters. You need to know these guys. Please study them. Flashcards, flashcards, flashcards. Dopamine. Dopamine is going to be the neurotransmitter that helps one with some motor and muscle control, but it's also going to be one that really you're going to want to know it in reference to the disorders associated with it. Dopamine is going to be an overabundance in schizophrenia, and it is going to be lacking when we are talking about people who have Parkinson's. Remember, Parkinson's is going to be indicated by tremors, the inability to control muscle movement, and so again, dopamine makes sense there. Serotonin, think this is your happy or your mood stability type neurotransmitter. So when we're talking about a lack of serotonin, you're usually talking about things like depression. Acetylcholine always goes with Alzheimer's disease. Now, Alzheimer's disease does have a motor functioning issue, but this also happens to be related to memory formation. And again, that's usually where they hit on for the AP exam, acetylcholine and memory. Endorphins, these are our naturally occurring painkillers. So when we talk about endorphins, they're often mimicked by opiates like heroin, morphine, etc. But these are the things that help us maintain or control pain. So they are these little kind of bursts of pleasure that help us kind of forget about pain or at least minimize pain. They also get released in times of actual happiness and pleasure. GABA. So when we're looking at GABA, this is your primary inhibitory neurotransmitter. This helps dampens other sending or stopping of other sending of messages. And glutamate, if you're sending dopamine, you're not sending a ton of dopamine, you're sending dopamine and a ton of glutamate. Glutamate is your primary excitatory neurotransmitter. It enhances the dopamine message. So you want to know glutamate, primary excitatory, GABA, primary inhibitory. Students previously said GABA, they just remember goes against brain activity. It doesn't really, it just inhibits it. So keep those together. Vesicles house neurotransmitters. Receptors are going to be the lock that those keys fit into that stimulate a message, and reuptake is when they go back home, they go back into their vesicles. You need to know the difference between an agonist and an antagonist. Agonist and antagonist, those are drugs. They are not neurotransmitters. They are drugs that mimic your neurotransmitters. So an agonist acts like, and an antagonist acts against. So an agonist will act as if it were an endorphin and help with pain relief. An antagonist would block the endorphins and block the pain relief. Now, you might be asking why you'd want that. Who cares at this point? Just know that that's what they do. All right, so we got through the neuron a lot faster than we did in class. And now we're going to talk about basic nervous system. Now remember, you have your central nervous system and your peripheral. Central goes down the center of your body. Peripheral, just like the periphery of your paper, is to the outside. So central nervous system, brain, spinal cord. Peripheral nervous system, this is going to be your autonomic system and your somatic system. Your somatic is going to deal with your reflex arc. So that's going to have to do with your efferent and your afferent neurons and your inner neurons. So if you go all the way down to the bottom of the page, you see types of neurons. You have your afferent that allow you to affect a change and your efferent that allow you to effect a change. Here's what I'm talking about there. Afferent is your sensory nerves that allows you to bring the information to your spinal cord or your brain, whichever happens to be the closest. And then your inner neurons process it. Inner neurons are the ones that are in your brain and your spinal cord. They interpret the message, and then they're going to send a message back down your efferent neurons, which are motor neurons, and they will allow you to yank your hand off of that hot stove or bring your foot up off of that tack that you're about to step on. That is your somatic system. That is your reflex arc system. Then you have your autonomic nervous system, and that is going to be subdivided into your sympathetic and your parasympathetic. This is your fight or flight system. This deals with your internal glands and organs. So if I'm telling you there is a tiger charging you, you are probably going to sympathize with that by freaking out and running for your life. Your heart rate's going to escalate. Your breathing's going to escalate. You are probably going to stop salivating, because who needs to salivate if you're about to die? You are going to take in as much light as your pupils get as big as saucers. All of that is going to be your sympathetic system kicking out. Now you realize that that tiger is, I don't know, a delusion or something, and you bring yourself down like coming down with a parachute. It's not an immediate off, it slows everything back down. And since a lot of this is working off of hormones and not just neurotransmitters, it has to run through your circulatory system. So you bring yourself down a little bit slower. Just think of any time that you've almost hit something with a car. You don't just immediately stop shaking. You have to kind of pump the brakes a bit. You kind of have to slow your system down. That's your parasympathetic, much like a parachute, bringing you back down to your baseline. Your endocrine system is ruled by your pituitary gland. Pituitary gland lives in the brain next to the hypothalamus, and it is going to control all of the other glands of your body. It also spits out your growth hormone, so that's a nice little kind of additive benefit of this gland, but it is the master gland of the endocrine system. The endocrine system is your glandular system. This is what spits out hormones. So testosterone, progesterone, estrogen, adrenaline, all of those are hormones. Those are all guided by the endocrine system, and they travel through your body using your circulatory system. Okay, time to take a breath. Now we're into brain mapping. And remember, I told you guys that there are certain ways to remember some of these things. An EEG is the electrical activity that is measured. And so with an EEG, it's a tight neoprene cap that is going to literally just tell you where electricity is being generated by the brain. It is good for sleep studies and for epilepsy. Outside of that, we're not going to be able to really tell you where or what is going on in the brain, but it helps us understand brain waves. CT scan, also known as a CAT scan. Now remember, you can see a cat, so a CAT scan is structural. CAT scans use x-rays. It's a basic, simple x-ray of the brain. A PET scan, you can pet a cat. So this is going to be functional. This will give you a color-coded map of the brain. The PET scan is going to use radioactive glucose, and that is going to be what is picked up by the x-ray. MRI is going to be structural. It's going to give you that nice slide-by-slide detailed view of the brain, where an fMRI uses oxygen consumption, and those magnets will pick up on that, and that'll give you that detailed functional view of the brain. The only other thing that's really not on here that you might want to know is TMS, which is transcranial magnetic stimulation, and that is a way to dampen parts of the brain, but we will talk about that, I believe, more when we get into therapy. But this helps us create virtual lesions of the brain. Here's our lovely neuron again. So I have this titled as DCBA, dendrites, cell body, axon. That is the direction that the message always travels. Dendrites receive the information like a lock and key. It stimulates them, sends a message to the cell body or the soma where it has that graded kind of potential. When it meets its threshold, it activates an action potential in the axon. It pushes sodium and potassium ions in and out of the cell body, causing a wave-like motion which shoots the electrical message all the way down to the very end of the axon where the neurotransmitters are housed. It pushes the neurotransmitters into the cell wall. They rupture, neurotransmitters spill out, they go to the next dendrite, they stimulate that dendrite, then they get released and they come back to the axon terminals where they go back home. The entire cell resets, and there you have it. Now we're in the brain. Let's see if I can get through this rather quickly. So 3 major structures of the brain: hindbrain, midbrain, forebrain. 3 major structures, or divisions, let's say. So hindbrain, 3 structures to the hindbrain. You have your medulla, medieval death. This is going to be your basic life support. This is respiration, this is blood circulation, this is yawning, sneezing, coughing, anything that you do without thinking. That's your medulla. So if we sever that, you're dead. Your pons just a little bit above your medulla. That is going to be sleep and arousal. And so I always told you guys you would never want to fall asleep in a pond, but another student came up with an acronym, perfect one night sleep. Either way, your pons helps with sleep and arousal. It also connects the cerebellum to the brainstem. That is secondary as far as what you want to know. Cerebellum, this is a big one. It's the little brain, and we keep finding out that it has more and more purpose to it. So the cerebellum is going to be balance, your kinesthetic sense, your procedural memories. It is going to be that little kind of bowed out part at the very base of your brain. It is going to help you with fine motor control. So if you can play a guitar and you can move your fingers in that really kind of fine coordinated fashion, that's your cerebellum. If you can remember how to tie your shoes, that's your cerebellum. If you can walk in a straight line, that's your cerebellum. So cerebellum is going to be the thing that keeps you balanced and coordinated. Move on to our next section of the brain. We're talking about the midbrain. Midbrain is going to be the area of the greatest neurotransmitter production, specifically dopamine production. That's important. Also, one structure of the midbrain that you need to know is the reticular formation. The reticular formation is going to run through the brainstem, and it's going to terminate just inferior to the thalamus. That's just if you're looking at a diagram. So reticular formation has to do with sleep and arousal. I said that about the pons also, but this one is more like your on-off switch for sleep and arousal. So if you haven't slept in a very long time, this will put you out. This will put you into a micro sleep. This will be the thing that turns your brain or you off kind of technically for a bit. So any damage to this, you would lapse into a coma. Then we get into the forebrain, and the forebrain is the big mamma jamma of it all. Forebrain, we're going to start with the thalamus. The thalamus sits right kind of on top of the reticular formation at the very end of the brainstem. This is going to be your sensory switchboard. It lets all of your sensory information know which proper area of the brain to go to, whether it's a visual information coming to the thalamus, back to the occipital lobe, back to the thalamus to be processed in the parietal or the temporal lobe. It is going to be the great orchestrator in the brain. The only sense that does not go through the thalamus, sense of smell. Then we get into your limbic system. Limbic system surrounds the thalamus. It has your hippocampus. Hippo never forgets. Or your hypothalamus. This has to do with all motivation. So hunger, sex, thirst, that is your hypothalamus. Body temperature, hypothalamus. And then your amygdala, this is going to be anger, aggression, emotion. So not just the bad emotions, but your amygdala is your emotion processing. So memory, sexual, hunger, thirst, body temperature, motivation, hypothalamus, and then emotion. Then we get to the forebrain, and this is everything in what we call the new brain. It's the cerebral cortex. Your cerebrum is going to be that little thin layer of neurons that kind of fits like a swimmer's cap on top of the cerebral cortex. First part, we're going to talk about frontal lobe. Frontal lobe, literally in the front of your head. So when you're thinking and you're rubbing your forehead, that's because you're touching your frontal lobe. Not technically, but go with that one. Your frontal lobe is going to be your primary executive. It is going to be the decision maker, the planner, the orchestrator of your kind of life. It includes your motor cortex. That is your choice motion. So if you're a hand talker like I am, I choose to move my hands, motor cortex. If you choose to walk forward, motor cortex. Now the fact that you're not falling over, cerebellum with motor cortex. Broca's area. Right now I am moving my mouth. The fact that I'm moving my mouth to produce words, Broca's area. Motor cortex, moving my mouth, Broca's area. Those two are in the same area of the brain. They both involve motion. Parietal lobe. Parietal lobe is going to be at the crown of your head. It is going to have your somatosensory cortex. That is your sense of touch. It is right next to your motor cortex. So your ability to move and your ability to feel are butted up against each other. Your parietal lobe also has to do with 3D mental rotations, your ability to manipulate and move objects in space. It also has to do with mathematics and music. It's a pretty developed area of the brain, but we're really concerned about our sense of touch and 3D mental rotations in space. Temporal lobe. Find your temple. Your temple is near your eyeballs or your ears. Well, our eyeballs are going to be processed by the occipital lobe. So let's just go with temporal lobe next to our ears. That's what we want to remember. Temporal lobe, hearing, primary auditory cortex. Wernicke's area is in the temporal lobe. Wernicke's area, comprehension. I have to hear it to understand it. I hope you guys can all hear my dog barking. Then we get to the occipital lobe. Occipital lobe, very back of the head. That's vision processing. Wonderful thing, the corpus callosum connects these two hemispheres. We have a left and a right. They can actually work independently of each other. We connect them with this bundle of nerve fibers. That bundle right there allows them to communicate, so you are neither right-brained nor left-brained, even though you have both, because they work together unless you are split-brain, and that's when we've cut it in half. Plasticity is going to be the brain's ability to modify itself. Your brain remains plastic as you age. It just becomes less plastic. Okay, so it means nothing to you guys, but I just paused to yell at my dog. All right, so plasticity, your brain is very plastic and malleable as a child, meaning that brain damage, while it's still going to have an impact, there's a greater chance to recover from it than in adulthood. Term aphasia, you should just know in case it pops up in a question, impaired use of language. Broca's aphasia means that you have the inability to move your mouth to form language. Wernicke's aphasia means that you can't comprehend language. Lesion is intentional brain destruction or tissue destruction of the brain. All right, so last two things. Split brain procedure. Literally sever the corpus callosum. Information that is going to come from the right side of your body will be processed in the left hemisphere of the brain. Information coming from the left side of the body, right hemisphere of the brain. Language is processed in the left hemisphere. Broca and Wernicke's area, left hemisphere of the brain, meaning that you have to have information coming in from the right side for you to be able to speak about it. If you have questions about split brain, you need to come talk to me. Hemispherectomy, this is going to be utilized when we talk about people who often have very severe epilepsy. We remove half of a hemisphere of their brain. That would include the frontal lobe, parietal, temporal, and occipital. It'll be removed. The underlying structures like the limbic system and the thalamus would not be touched, but we do this to help with epileptic seizures. Again, these kind of surgeries are going to be done when someone is very young, the hemispherectomy, and they can remove the left or the right. But what we often find is that the abilities that tend to be localized to one hemisphere versus the other will then later grow upon the other hemisphere of the brain, but only if the procedure is done to a very young child. Important people for you guys to know Phineas Gage. He is going to be the guy who had the tamping iron shot through his head, and really what that is historically it gives us this idea that one, there are different areas of the brain with kind of specificity, different areas that have kind of important places or structures to them. And then Gazzaniga, he was going to be, him and Roger Sperry did the split brain procedure, but again, don't worry so much about Gazzaniga. Definitely no Phineas Gage. I believe this is it. If there's a next slide, I will apologize, but other than that, that should be a really fast review of the brain and the neuron. Okay, last little bit, not brain neuron. This happens to be genetic influences. Not really much to talk about here. The only thing that I really want you guys to focus in on all of this is twin studies and adoption studies. Twin studies, we look at them because we can look at the exact same DNA in identical twins, and then dizygotic or fraternal twins, they have the exact same first environment. Adoption studies, very important because we can look at a child and see what they have in common with their biological parents versus their adoptive parents similarities, environmental, biological parents, that is going to be genetic. The rest of this is really kind of terminology that you should have a general idea about. Don't worry so much, but also remember that when we look at brain development, It is not just genetics, it also is going to be environment. So tutored human enrichment, any of the studies where we've looked at children in isolation or neglect, all of those things become very important. So general idea about here, but again, if you guys have any big questions, please come see me. This tends to be stuff that you've had largely in biology and should be a slight review for you. That should be it, and hopefully I can speed talk my way through your next unit review. Bye guys.